This provides a great segue as we go into the next talk that'll be given by Dr. Denise Sanger, ecosystem scientist at Marine Resources Division of South Carolina, DNR, and she will talk about the environmental and habitat quality of the Port Royal Sound.
Good morning, everyone. I'm really excited to get to talk about Port Royal Sound and the overall environmental quality. And so I do want to recognize a lot of this is based on long-term data that's been collected at DNR for many years, but the Port Royal Sound Foundation provided funds for us to be able to actually synthesize it at the Port Royal Sound level. So very exciting. Oh, there we go. Thank you. Technical difficulties. So I do want to highlight some different habitats because it's going to be important as we proceed through the talk today. And so the one thing you notice is lots of open water, or sort of the light blue color, as Andrew talked about. And so in order to then look at some additional habitats, we're going to focus in on the Okatee as our example. And so in addition to that light blue color, which we consider again to be what the CCAP program terms open water, it's really less than 100 meters wide in width. So it's pretty much any water, right? Whether it's in a creek system, a river system, or sort of the open Port Royal Sound, that's less than 100 meters wide. And so you can see where that occurs here on the Okatee River. We also have our large tidal creeks. And so that we define as anything that's about greater than 10 meters wide up to that 100 meters wide. And so you can see that in that darker blue. And then finally, Andrew talked about the very headwaters or small tidal creeks. And so I'm just showing you 3 here shown in brown that we have sampled in the past. And so these are really getting us to that really direct connection with the land. I'm not going to really talk about salt marshes today, but they are, such a significant estuarine habitat that I wanted to make sure to at least capture them. So I'm going to focus on a program called the South Carolina Estuarine and Coastal Assessment Program, or CCAP. And this is a statewide program where we have, since 1999, sampled over 800 sites. And so we focus in on those open water and those tidal creek sites, and we actually sample 30 sites, 15 open water and 15 tidal creeks each year. And so this provides us a really great opportunity to then be able to focus into the Port Royal Sound watershed. And so the circles are the sites that we've sampled in the open water areas, and there have been over 100, or there have been 156 since 1999. And then the triangles are the large tidal creek sites, and there have been 123 that we have sampled. So we really can focus in on these watersheds and be able to say something about the proportion of habitat that gets classified differently. And so I also want to highlight, right, we have these 2 time frames that we can then look at since we have this significant data source. And so we have sort of a first 11 years shown in green and a second 11-year time period shown in pink. And so how do we do our environmental quality assessments? Well, it comes down to looking at water, sediment, and biological quality to get at overall habitat quality. And so we have primarily looked at dissolved oxygen, pH, salinity, the nutrient levels, chlorophyll levels, and fecal coliform levels within the water. For sediment, we actually send, the picture represents a grab sampler that we send to the bottom. It scoops up mud, it brings it to the surface, and it enables us to then actually look at what does the sediment quality look like. And in this case, we're looking at chemical contaminants, organic carbon in the sediment, and the toxicity of the sediment. From a biological quality perspective, we really are focusing in on the animals that live in the sediment. We call them macrobenthic organisms, and they really are fish food. And so I'm showing you a few examples. The crustacean is a small amphipod. You can see a clam, and then you see lots of worms, which are the primary food resource for lots of fish and crustaceans in the estuary. And so that gets us, right, we can take all those pieces and we can then do a habitat assessment. And so to give you some idea of how we do this, I'm just showing you 2 sites as examples. And what you can see is, so for each of the different metrics, right, we go ahead and give it a score. And so in the first site example, you have water being good, the sediment being good, and then the biological quality being fair. That then comes together into an overall score of good. If we look at site number 2, for water, this one got classified as fair. For sediment, we ended up with a poor quality assessment. And then for biological, it was again green or good, which when you wrap that together, you end up with an overall habitat score of fair. So now let's talk about, right, we took all those sites, we classified them, and now we can actually take that information and summarize it into an overall assessment of the Port Royal Sound watershed. And so I'm going to show you for all these pie diagrams, I'm only going to show you the most recent 11-year period, so that 2010 to 2020. And the water quality is the most complex assessment that we do. And so I want to spend a few seconds walking through it with you. And so you've got 3 different metrics. You've got total nitrogen, total phosphorus, and chlorophyll, so those bottom 3 pies. And essentially you're looking to see what the proportion of the pie is green for good, yellow for fair, or red for poor. If you see black, it means there was no data collected for that. So that we take those individual sites, right, they get wrapped up into a eutrophic index. We can then take that eutrophic index, combine it with the fecal coliform data, the dissolved oxygen data, and the pH data to come up with a water quality score for the open water habitats, which will always be on the left. And so the entire, the open water in Port Royal Sound ends up with a quality score of good. If we now look at the large tidal creeks, right, you really want to key in on how much green is there, right? And the fact that we start to see more fair and poor habitat quality or water quality. And that is largely due to the closer connections with the land that Andrew talked about. And so, but when you look at the overall 2010 to 2020 time period, you can still see that it's, you know, about 70% good, with the rest being fair and poor. If we look at the sediment quality, again, we've got open water first, and you can see that, right, overall, really lots of green. So overall, 100% of the sediment quality in the Port Royal Sound area is considered to be good. When we add in those tidal creeks, we find that you start to see, again, that more of that yellow, tiny bit of red, but overall, still really good sediment quality. If we now look at the biological quality, and this is just one metric, but it actually involves numerous different types of organisms that all get combined to develop that one metric. And so in this case, you again see, right, a little bit of fair this time for the biological quality in the open water habitats. But if we look at the large tidal creek habitats again, we start to see more of the fair, a little bit of the poor, but still about 75% comes out as being good. If we now take those 3, water quality, sediment quality, and that biological quality, and we wrap it together, we see that 100% of the habitat in the Port Royal Sound watershed is considered to be good. If we add in those large tidal creek sites, and we wrap everything together, again, about 75% ends up being good quality, but you start to see, you continue to see that little bit of yellow, or a little bit of red, and more so the fair habitat. And so remember at the beginning, I told you we had these 2 different time frames. And so we can begin to look across these 2 time frames and see whether or not we see differences as well, or changes. So in this case, again, you're looking at the open water habitat, and it's very similar to the pie graph because you're just on a 100% basis. And so you can see that in the sort of early 11 years of CCAP, there was a little bit of fair habitat quality, but in the 2010, 2020, it was all good. The tidal creeks, we again see sort of the same pattern of more, fair, and poor. And in this case, we actually see some additional fair and even some poor habitat quality in that more recent time frame. So what else can we do with CCAP? And so again, since we have each of those individual sites, right, we can do that proportion like I've just talked to you about, but we can also go in and say, are there particular areas where maybe we should be focusing, or potentially areas that, you know, require further exploration. So I've gone back to that full watershed area, and I've only plotted up those tidal creek sites in the triangles. And again, we're going to focus in on the Ocoee River and the Ocoee headwaters, where this particular site scored as yellow for the overall habitat quality. And then we can go in and say, well, let's see, for water quality, it was fair. For sediment quality, it was good. And for the biological quality, it was poor. And so again, we can begin to then look even further down to figure out which of the metrics were driving these patterns. So lots of potential there for further exploration. Now I want to turn my attention to the small tidal creeks. And so this is work that was started back in 1994. It is not sort of a continuous long-term monitoring program, but has sort of been developed at different time frames to answer different questions. But the overall question is largely the same. What is the relationship between watershed parameters and the environmental quality in our headwater tidal creeks, or small tidal creeks? So again, you're looking at the very headwaters to the Ocoee River. And this is a site we have sampled over many years. And I've gone ahead and defined its watershed. And the red is similar to what Andrew showed you. It is the impervious cover level. This is a layer from 2011, just to give you a perspective. Currently, in the 2016 data set, the impervious cover in that watershed is 32%. And so I'm not going to show you data to back this up today in the interest of time, but I will sort of walk you through. What we've done is we've developed a conceptual model after looking at the parameters in the watershed, right? Those are what we call our stressors, our coastal development activities. And the best indicator for us has been the level of impervious cover or impervious surfaces. We then go in and sample each of those tidal creeks to get an assessment, those small tidal creeks, to get an assessment of what does that water and sediment look like. And so that's where you get at the physical chemical changes and is the exposure. And so we do see things like altered water quality and hydrography, increased microbial contamination, those fecal coliforms, and increases in chemical contamination in the sediment. These changes in the physical chemical environment, right, can elicit a biological response or an impact to your living resources. And you can see reduced biological productivity, altered food webs, and impaired animal health. And then more recently, we've tried to add on sort of the societal component. How does the health and well-being look from a people standpoint? And so things like, do we see increased beach or shellfish bed closures, increases in flooding, and the sort of public health risk component. And so we have been very fortunate through the years that we have collected enough data that we can actually develop cut points at which we think once you've developed that headwater watershed past a certain impervious cover level, that you will see a change in your physical chemical environment. And that's at about 10 to 20% impervious cover in the watershed. At what point do you then see a biological response? It's at about 20 to 30% impervious cover in the watershed. So these numbers are in very good agreement with research that's gone on in freshwater streams around the country. And so the last thing I want to show you is an example when Andrew talked about changing the fact that we're altering the surface water leaving the site, we're increasing that, and it comes down to the rate and volume that it's coming in. And so here's an example from Rosedew Creek, which is at the headwaters of the May. On the left, you have an image from 1994. It is, the trees are red because this is what's called an infrared image. And then as we've progressed in technology, we now more commonly get true color images, which is what you see in the 2013 photograph. And so again, these are those very headwater areas. And you can see 46 is the road at the bottom. So if we sort of zoom in to these two particular areas, what you can see is that on the left in the 1994 image, you see a lot, and I've gone ahead and highlighted the actual creek channel with yellow. You see a lot of switchbacks, right, and a very meandering tidal creek. In comparison to the 2013 image, where you actually start to see a lot of those extensive curvature gets cut off, and the water flow essentially is becoming a straighter path through the system. And so you are straightening channels. And we've seen this in a number of other areas as well. And so what changed in this 19-year period? Well, there was a change in 19% impervious cover in Rosedew's watershed, and we saw an increase of 30 stormwater ponds in this system. So again, exemplifying that change in rate and volume of runoff. And so in summary, I want to highlight the fact that, right, the majority of the Port Royal Sound quality is still classified as good, which is wonderful. We do see sort of more impaired quality once we start moving up into either large tidal creeks or even to those small tidal creeks. And really that's due to the fact you have a closer connection with the land. Most historical studies in like the national level are done solely in those open water habitats. But when Bob Vandola started this project, he was a close friend of Fred Holland, who started the Small Tidal Creek project. And they started recognizing the fact that if you're going to see that close connection, right, we really need to move into those smaller systems. And it certainly has proved very fruitful. Level of development is relatively low, but right, it's continuing. And so some of the watersheds, an example I gave you was the headwaters of the Okatee at 32%, right? That's getting into that potential for biological impairment level. We do, again, have the opportunity here to focus in on some areas of concern for further study. And I don't think we've reached a tipping point to sort of broader impairment. You're certainly not seeing it in those open water areas. And so there's a great chance to continue to keep the quality in Port Royal Sound really good. And I wanted to come back to the salt marshes here. And I think, from a research perspective, this is also an area in which a lot more information needs to be developed so we can better understand how our salt marshes are going to respond to things like sea level rise. And that's it.
Thank you, Denise. We have a couple questions. The first is, the question was asked about the open water nitrogen has a large no data segment. What is the cause of this, and what is needed to get more data?
So that is largely just due to analytical interference. And when you go into the higher salinity areas, you tend to get more interference with regard to actually measuring nitrogen levels. And so that's why that occurred.
And when you say interference, can you explain what you mean by that?
So when you actually get into the laboratory to essentially, I'll call it clean up the sample to remove that salt content. And so sometimes you just, depending on the analytical method, sometimes it can achieve nitrogen levels and sometimes it can't.
All right. Okay. Thank you. Next question is, can you delineate the way you broke out good, fair, and poor for DO and pH?
Ooh. I should have brought that.
I know those are analytical measurements, so you may not have them on your fingertips, but
right. Yes, and I could have created, I could have shown you the whole matrix. And so yes, each one of these thresholds were developed based upon a whole series of different data sets. And it largely depends on whether or not there were thresholds available from other sources that can be utilized, or at the time, Bob Vandola and his crew went through with the DHEC folks to figure out where might you actually set cut points based upon historical data. So sometimes it's solely based upon DHEC standards, like fecal coliforms utilizes the surface water standard, the swimming or recreational standard as the threshold from good to fair, and then you're actually, to get from fair to poor, it's utilizing the shellfish standard. And so it just, they were developed over time by looking at either historical data or utilizing existing thresholds. And so
Okay,
I
great.
don't recall exactly where pH and DO come in.
And we'll, as I said in the previous, with an earlier talk, for those of you interested in some of the more technical aspects, we'll have links to the full studies that have been done, so you can delve into those to your heart's content. Our next question is, what has been the impact of named storms in the last 10 years? I know this is not a stated purpose for your CCAP research, but is there any qualitative observations or casual observations that you made that might relate to that?
Right. So one of the things that we sometimes see is in most of our, so we typically do our statewide assessments in order to get the number of samples we need to look at the proportion of habitat. We do a 2-year assessment. And so in the past, we have tried to look at sort of what was the either preceding rainfall around the summertime or the actual sort of salinity levels. And sometimes we see relationships with salinity and rainfall, and sometimes, but as like the years go on, then sometimes we lose those relationships because they're not, linear. And so we have not looked at the 2019-2020 data set again, but we always try to see whether or not we're seeing some clear relationships. And we also, I almost want to go back to Andrew's. So Andrew did a synthesis statewide with a graduate student looking at more climatological patterns with the environmental quality. I don't recall it off.
More contaminants the following summer after a wet winter.
So more contaminants the following summer after a wet winter.
Okay, so one of the we think of big storms, but these El Nino winters where we get a lot of rain but the trees are not leafed out, actually sounds like they're making a significant impact or a measurable impact based upon what you're saying.
They have the potential to, yes.
They have the potential to,
analysis.
Yeah. Yep. Okay, great. Next question, and this may be beyond your scope and something that the Port Royal Sound Foundation Spring Island Trust have been discussing as a topic for the next State of the Sound, but I'll go ahead and read the question and we can maybe touch on it as we go through the rest of the symposium. Is, can something be done with stormwater ponds to make them more effective in slowing the storm flow? I'm going to go ahead and comment on that, since I've been working with the municipalities, and the key thing there is that many of these stormwater ponds are viewed as amenities, being an aesthetic pond for the residents that live around these ponds, and therefore there's strong pressure on the general managers to maintain those ponds at a high water level for aesthetic purposes, which is actually destructive from a water quality standpoint. The ability to keep pond levels lower so that when they get a heavy rainfall event, they can actually hold on to the water rather than it just flowing through is what your goal is. However, many of these ponds were not built with water control structures, and that includes some of those in the Bluffton area and some of the older ones. So if you had the financial resources and could go back and put in a water control structure where you can maintain water depth and keep them low when you wanted to, that would be the ideal solution. But that's there's a financial goal or block there that makes that difficult. The last question we have here, I think, is what are the key factors bringing down the quality in tidal creeks versus open water? Is it the proximity to development in communities?
that's part of it. Yes. You know, they really have that close connection to the land, but naturally tidal creek systems are more stressful systems than your open water habitats. And if you think about, you know, it's less water in that area, and you have just natural oxygen demand. And so, you know, one of the things that we've always talked about is the fact that, you know, we really think tidal creeks and salt marshes are such good nursery habitats, because particularly as you move upstream, because they are such a stressful environment naturally, that you'll find that you'll keep bigger organisms, right, who want to prey upon the smaller ones out. So you have to very much, you know, take into account the fact that as you move upstream, they are much more or naturally stressful. But then you can tease apart, you know, when do you actually start to see the impact from development. So
it's a combination of both the natural conditions and the human caused factors. So,
right.
In other words, are you saying some of the values, if somebody's used to working in freshwater streams, for example, dissolved oxygen and all, that those indicators of what is healthy freshwater creeks those measurements may not apply to tidal creeks, because even without the impact of humans, tidal creeks are going to have lower DO and more extreme conditions. Is that correct?
Correct. We consistently see hypoxic conditions in headwater tidal creeks. And, you know, you can start to see lower oxygen levels in your large tidal creeks. And then, you know, by the time you get back out into that open water habitat, most of the time, you know, your concentrations of oxygen have gone back up. So.
Okay. One last question. Does the depth of the water also play a part in how you determine what they are called, or is it just how close to land the water is? I believe this may relate to tidal creeks versus headwaters.
Right. And so one of the things I didn't say is when we sample large tidal creeks in open water, we're always doing it by boat. And so, you know, that designation was based upon, you know, can you actually get vessels into those areas? And so that's how, you know, that fell out. In the headwater areas, we actually sample by foot. And, you know, sometimes we can take a canoe with us and sometimes we can't. It just depends on the system. And so from a headwater perspective, we consider the bottom end of a headwater tidal creek to be roughly a meter deep at low tide. And so, you know, if you look at a topographic map and you, most of the time for large tidal creeks, you're going to always see sort of a shape or an outline to that water body. But when you finally get to just seeing a single line that designates a system, that's typically what we end up calling a small tidal creek or a headwater tidal creek. So it's just kind of naturally worked out that way. But that's sort of the distinction.
Well, thank you very much, Denise.